So what we're going to try to do today is extend that discussion yesterday of the secondary structure, the idea of the alpha helices and the beta strands, which we referred to yesterday as the beta pleated sheets. And I'll review this with a couple of slides. And then once we get through these concepts of the secondary structure, we're going to drill down and start talking about tertiary structure. Now, it's true that there's a certain level of three-dimensional structure or shape which derives from the relative regions of alpha helix and beta strand. But to get the really proper functional three-dimensional shape of a protein, one requires the R groups on the amino acids. And what we're going to emphasize here is the idea of some of those R groups being hydrophilic, while others are hydrophobic. And the hydrophilic and hydrophobic nature of the R groups now will impart certain function and certain structure to these proteins. So we'll try to work our way through the combination of secondary structure and tertiary structure to get to a three-dimensional shape for a protein. But all of this is going to depend upon the idea of having that protein within an aqueous environment, living surrounded by water. Then we have to move one step farther, because you'll remember one of the major ideas we introduced was that a protein can, in some cases, span the lipid bilayer. We'll find out in great detail how this gets done at the cell biology level, but for now, recognize that simply there are some transmembrane proteins that are forming the basis of the lipid mosaic model of the membrane. We'll run through this in such a way as a single pass and then a multipass transmembrane protein, and then talk about how they're going to generate a channel. One of the things I want you to think about as we work our way through this is that if we're going to have a single alpha helical region which goes across the lipid bilayer, that the amino acids that are embedded in that hydrophobic region of the lipid bilayer had better be hydrophobic. But that if we have a single pass transmembrane protein with only hydrophobic amino acids facing the fatty acids, we can't very easily then generate a proper channel, a proper hydrophilic pathway from the inside to outside of the cell. So in order to generate a proper hydrophilic pathway, we now have to thread these amino acid sequences across the membrane several times. One of the restrictions we're going to see in eukaryotic cells is that these transmembrane regions can only be alpha helices. The rules are we can't have beta sheets crossing the membrane. There may be a few extreme cases in prokaryotes and bacteria where you can use beta sheets to cross membranes, but not in eukaryotes. So now we have to constrain ourselves to using alpha helices going across the membrane to make a channel. We'll talk about how that gets done. If the first period predicts what we'll see here, we probably won't get as far as eukaryotes and prokaryotes. But one of the things we're going to spend some time on in here is distinguishing eukaryotes from prokaryotes, particularly recognizing that prokaryotes evolved first. They've probably been around for close to 3 billion years. And they can be broken down into the eubacteria and the archaea. One of the hallmarks of all of these guys, whether they belong to this group or this group, is that they are single cells. They may get together in colonies, but they don't make metazoan kinds of animals. To become a multicellular creature, one has to invoke the idea of a eukaryote. And the eukaryotes, in fact, have done something really clever. And these guys came up, someplace between 1.5 and 2 billion years ago. So they've been around quite some time. And what they were able to do is they were able to figure out a way to make membrane-bound compartments. They specialized function in different membrane-bound compartments, and through that specialization, these cells were able to fit whole new niches. And so they were really an important aspect of how life has developed on the planet. And we'll talk in some detail about the specializations of these different compartments. The first place we'll talk about that is that one of the major discrepancies or differences between the prokaryotes and eukaryotes is that the DNA in the prokaryote is a simple circle. It exists in the cytoplasm itself. But once you get to eukaryotes, one of the most important of all of the compartments is that we sequester the DNA. We sequester the material which contains the instruction set for making proteins. We sequester that information into the kernel, into the karyote, into the nucleus. And we'll now store it at that point. And we'll see that not only do we store all the DNA into the nucleus, but one of the hallmarks of being a eukaryote is the DNA is divided into chromosomes. And the chromosomes in prokaryotes are simply circular. But having said that, one of the major ideas we're going to find among these two is that at least biology as we know it today, it may not always have been this way, but all of biology that we know it today obeys some certain rules that we call the central dogma. And that is, we have an instruction set in the DNA. We then take that information instruction set in the DNA, transcribe it into RNA. Then we take the information in the RNA and make it into protein. That's true for all biology as far as we know it. It may be that a couple of billion years ago it was different, but now that's the way it works. And that's true for both the prokaryotes and the eukaryotes. There's some subtle differences between these two, but nonetheless, that's the major idea of the central dogma. Then finally, if there's time, we'll finish up with the idea of histones organizing the DNA.